The circulatory system is responsible for transporting gases, nutrients, hormone, cellular waste products throughout the body, comprising three important components: blood, heart, and blood vessels. Blood is a connective tissue composed of blood plasma and formed elements. Whatever blood is in liquid form inside the blood vessels, but what is its condition if it comes to outside in case of injury or damage, which is related to the current topic. As long as the blood remains in its blood vessel, it is in liquid form, but if it is drawn from body, it thickens and forms gel, which is clot, which consists of insoluble protein fibers called fibrin, in which the formed elements of blood are trapped, and the process of gel formation is called clot. The fundamental reaction in the blood coagulation is the conversion of soluble plasma protein fibrinogen to fibrin to form clot. If we consider the procoagulant and anticoagulant, then during normal condition, procoagulant is non-functional and the anticoagulant is predominant. But during vessel damage, the procoagulant is dominated, and the activation of different factors resulting in blood coagulation. Clotting done by three mechanisms, where if blood vessel rupture or any damage initiate, a cascade of chemical reaction induce formation of prothrombin activator, which activate the inactive prothrombin to active thrombin, and then thrombin activate fibrinogen to form fibrin, which forms fiber. Thrombin activate fibrin stabilizing factor, which help in cross linkage of fibrin, which ultimately form the clot. This reaction you can see. Fibrin monomer hold together generally by non-covalent hydrogen bond, but converted to covalent bond by the fibrin stabilizing factor. Prothrombin or factor II, a plasma protein produced by liver, and for its activation vitamin K is necessary. That's why deficiency of vitamin K causes significant bleeding, trauma to vascular wall or to blood, induce prothrombin activator formation. These are the blood clotting factors. Most of the factor you can see is formed by liver. The factor 3 from damaged tissue or platelets. Factor IB or calcium ion, which from diet, bone. Factor V and XIII from liver and platelets. There is no factor called VI. You can see the name of the factors as well. Like factor I called fibrinogen, factor II called prothrombin, factor III is tissue thromboplastin, factor IV is calcium ions, and factor V proaccelerin. There is no factor VI. II is proconvertin, factor VIII is anti-hemophilic factor A, factor IX is Christmas factor, factor X is Stewart factor, factor XI is anti-hemophilic factor C, factor XII is Hagman factor, factor XIII is fibrin stabilizing factor. The clotting cascade is generally triggered by two pathways, extrinsic, intrinsic. Do you know why extrinsic pathway is so named? Because tissue factors is a tissue protein which is called tissue thromboplastin or factor III is released by damaged tissue or platelets, comes into blood from cells outside or extrinsic to blood vessels. This pathway is generally shorter and occur rapidly. The chemical reaction you can see including release of tissue factor, the factor X activation, and then activated factor X induces the prothrombin activator. And by a series of chemical reaction triggered by tissue factor, Finally, a clot or gel forms. In the next slide, we will see the detail of chemical pathway. So, if we discuss about the detailed reaction of extrinsic pathway, then when injury or any tissue trauma happens, then the release of tissue factor happens, which activate the factor VII. The VIIA means the activated forms of VII. The VIIA then help in the activation of factor X. Ca++ also plays an important role in the activation of factor X. Factor X then help in the formation of prothrombin activator. In this step, also calcium ion plays a vital role. Prothrombin activator help in conversion of inactive prothrombin, which is factor II, to form active thrombin. The conversion also induced by platelet phospholipid. Now the thrombin help in activation of fibrinogen to form fibrin. The active fibrin monomers cross-linked to form the clot. The tissue factor also help in the formation of prothrombin activator. Next is the intrinsic pathway, and this is so named because the activators are either in direct contact with blood or within the blood. This generally occurs slowly, and the steps are the trauma caused the activation of factor XII and platelets phospholipid also released. XII factor activation, then activation of factor IX or antihemophilic factor B, then factor X or STRAT factor activated. Here VIII plays a vital role in the activation, which ultimately form prothrombin activator, and then the same as in the extrinsic pathway to form the fibrin clot. We can see the reaction in the next play in flowchart. There are the different factors name used in the process. The trauma cause activate the factor XII, which activate factor XI, or Hagman factor, which activate Christmas factor or IX. Thrombin activate factor VIII. The activated factor IX active VIII and calcium. Platelet phospholipid help in activation of factor X or STRAT factor. The latter reaction are same as in extrinsic pathway. Like factor X activate prothrombin activator, which forms thrombin. Thrombin activates fibrinogen to fibrin, which cross-linked to form clot. These are the name of factor used in intrinsic pathway. There are some reactions you must have identified in the pathway. So let's discuss about the crosstalk between two pathways. First is the intrinsic pathway. If any damage in the blood vessels activates the factor XII or Hagman factor, which activates factor XI, which activate factor IX. Activated IX, activated VIII help in activation of factor X, which activate prothrombin activator. Prothrombin activator activate prothrombin to thrombin, which activate inactivated fibrinogen to fibrin and ultimately clot. In extrinsic pathway, the injury caused the activation of the factor VII, which activate factor X. Both extrinsic and intrinsic pathway activate factor X, which help in the clot formation. This is a diagram for a research article you can see in the picture, where the common pathway area you can see. Anticoagulant. What is it? It can be defined as the substance which prevent the blood clotting. For example, heparin and coumarins. Heparin act as an anticoagulant by inhibiting thrombin activity, and coumarins prevent the blood clotting by interfering vitamin K action. And in previous slides, you can know how important role thrombin and vitamin K plays in the blood coagulation. You must have heard about hirudinaria, a genus of leech. Hirudin, an anticoagulant obtained from hirudinaria. Hirudin interacts with both fibrinogen binding and catalytic sites of thrombin, which completely inhibit procoagulant action of thrombin. In vitro, clotting inhibited by addition of sodium citrate and EDTA or ethylene diamine tetraacetic acid, which is a chelating agent, chelate or bind to calcium. And we know that the calcium plays a very important role in the clotting, so these chemicals prevent blood clotting. Hemophilia, a clotting disorder. We can say that inherited clotting disorder. This disorder is hereditary, means it transfers from one generation to the next. The gene is carried by X chromosome. It is X linked recessive genetic disorder. It is generally two types. Hemophilia A, caused due to absence of factor VIII, and hemophilia B, due to absence of factor IX. These are the references you can follow. That's all for today. Thanks for watching. If you like the video, please like, share and subscribe for more such valuable informations. Thank you.